So yeah, I've got three topics. The first one we're going to talk about is identifying your sequences after they've gone through ONT barcoder. Feel free to follow along with the software itself if you've got it installed, but at least two of them are web-based, so you could just go and play with it while we do it. Why do we want to check the sequence ID? I mean, it's kind of self-obvious, but I'll go through the list anyway. Quality control of your sequences before you upload them to Bold helps keep Bold clean. Bold is a workbench. It is not a reference library. All the meta-barcoding people in the world, including myself, treat it as a reference library. You speak to the people who have built Bold, and they say it is not a reference library. It is a workbench. It has millions of records of varied quality of identification, of varied level of identifications, but all of us in the meta-barcoding world kind of just throw that out of our heads and just go, oh, wow, look, it matched that. That's great. Move on, publish that species. So we're, as part of the BGE project, going to do a lot of reference library curation around Bold, and I'll talk about that in Ben version 3 a little bit later. But just, it's obvious, but please do check the identifications of your sequences before you stick them in Bold, because if we're doing all of this reference library curation, it kind of relies on people being good when they're uploading the data. Otherwise, you quickly pollute the data set again with bad stuff. And it could be human. Human is flagged in Bold, or bacteria and stuff like that. So Bold has its own checks, but well A1 and well B2 could be swapped around in the labs. And so it's those sorts of checks that you really need to be careful for. So there's sample misidentification. Occasionally, I misidentify insects as a morphologist. I know, shock horror. I'm sure I'm the only person in the world who misidentifies things based on morphology. But if I'm not the only person in the world who misidentifies things based on morphology, then occasionally the sequences differ from what the morphological expert has said, and maybe someone needs to look at that specimen again. Maybe you need to look at the things that's matched in Bold again, because of course it could be what it's matched is wrong versus what was identified. But basically, if something comes up that doesn't match perfectly, have another look. Yeah. I mentioned sample contamination between wells and extraction and PCR. We don't often see this thanks to multi-channel pipettes. It's quite hard to put things in the wrong wells. But occasionally it comes up, and I'll email Joanna, who does the tissue sampling in our project, and I go, this is coming back as a beetle, but in the database it says it's a fly. Could you go have a look at what's in the well and what's going on there? Obviously, sample failure, Wolbachia is prevalent in a lot of insect groups, human, fungal, whatever's in the PCR water or the non-PCR water that you're using for your PCRs and things like that. Funny story about that. My first ever new species of cicadas that I was doing in my PhD, I drew a tree. There were these really long branches. I went to my supervisor. I said, oh my God, I found a couple new species in this group. And he said, have you blasted these sequences? And I was like, no, they were bacterial contamination anyway. So don't do that. And I said Bold already has its own internal checks for like stop codons and things like that. The beauty of using ONT barcoder is we get full-length, proven, error-corrected sequences. So it's far fewer of those sorts of errors go into Bold if you're using ONT barcoder and ONT sequencing than if you were using traditional Sanger sequencing. So that's great. So there's several options if you want to identify your sequences. There's the GBIF one. If you want to do this now, just Google GBIF sequence ID and you'll find it. There's obviously the Bold workbench, so you can go to Bold. You log into that. You don't have to log in actually to do some IDs, but if you want to do a lot of IDs and you want to get the IDs emailed to you, you do need to log in. There's Bold Digger, which is the best named piece of software I've ever heard of in my life, written by Dominik Buchner in Florian's lab in Germany. And what that does in essence is quite clever, is it queries Bold for you, downloads the HTML file, the web page result, and then converts that into a spreadsheet and does other very clever things. And then there's Blast. I'm not going to talk about Blast today. You just blast your sequences. We can if you really want to, but I think these three cover the vast majority of the things we're doing, at least with CO1 barcoding. Sarah? Yeah. As well as the public one. Yeah, exactly. So yeah, GBIF, this one I'll go through it a little bit more detail, but GBIF can only get the public data. There is a lot of private data in Bold. Again, as part of BGE, we are trying to politely turn the screws on the people who have this private data and say, well, if you were funded by the EU. That really should be public data now, and try and encourage people to get that public data. Yeah. But yeah, just if you want to try, I'm going to go through each one of these and give you some pros and cons. So GBIF sequence ID, it's very fast. I'll caveat that with when it's working. You may go and try now, and what I've done is I've loaded the two leeches from our Ukrainian colleagues, and it was like, bam, there we are. 2 matches, excellent. I then loaded the FASTA file from the 115 or so barcodes that we generated, and it got stuck at 2 and stopped. And yeah.
Will we see those live to your computer also?
Yeah, I'll go through this, yeah. I've got 45 minutes, yes, Spiros. This is, whew. Times 3, exactly, yeah. I'm going to talk slow. You're going to be so sick of me by the end of it if you're not already. So anyway, yeah, it's really fast. It's web-based. You don't have to install anything. And one of the relatively good things, I guess, good or bad, is it like spits out the taxonomy aligned to the GBIF taxonomy. So if your group in GBIF is a good taxonomy, then you're happy. But if it's not, then yeah. Cons, it's only on public data, and I mean the current data set is March 2023, so you can imagine there's quite a few sequences have been added since then. It matches based on, they take the public data, I think, and then they do a 99% clustering on that public data, and then you match against those clusters. That is different to the way Bold does it, and that is different to the way Bold Digger does it. And you'll see, if you take the same file and run it through these different programs, you will get slightly different results. Typically what you'll get is one of them will match to a species and say, I have matched to this species, I'm happy with that. One of the others will say, I've matched to the same genus, but I'm not happy assigning it to a species, essentially. And yeah, it doesn't always work. Used to work brilliantly. I think we just need to tell whoever installed it at GBIF to maybe have a look at it. It's very straightforward. You upload a FASTA file, you download your results. So I'm not going to do a live demo. I'm not as brave as Amrita when it comes to these things. So we're going to just go through it. Here is the web page. You can either stick your FASTA file over there, or you can paste your sequences over there. Like, voila, look at that, one I prepared earlier. And this is what happens when you do that. It magically goes off and does a blast search against that clustered public database. And I had hoped we could have filled the screen, but it stopped at 3 out of 84 sequences when I tried this the other day. So I just said, well, for demonstration purposes, good enough. It's doing a blast, so it has all the regular values you get from blast, like bit scores and coverage and length and all those sort of things. It comes back with the scientific name of the match. It's got different types of matches, so like a close match here if it's below a certain threshold. In this case, it was a 97.8% match, so it says it's a close match. These are 99.7 and 100% matches, so it's an exact match. And then it spits out the taxonomy of what it's matched against. Imagine it's done all of them successfully. It gets to the end here, and it tells you how many it matched at each level. And you can then just go and download. In the bottom corner, there's a little download the CSV file. And so you'll get a spreadsheet with all of these matches. So that's that. It's really, when it is working, it's beautiful. And this is the spreadsheet output for that example run. Ignore the fact that this is all blank. You would normally get it filled out when it's working. And you can see here that that specimen G12, it's matched to an ichneummonid wasp in that genus, but it's matched it with a 99.7% identity. Okay, so we'll have a look a bit later. I have a feeling that when we query this against Bold, Bold will tell us what species that is and say, well, we have a record with the species name, so we're going to add the species name to that. So there's pros and cons to using this one. So yeah, jumping into Bold. Any questions about that? We have time, so stop me at any time. Negative, okay. So the Bold workbench, lots of pros, obviously. So you can do top 20, top 50, top 100 matches if you really want to look at how messy the underlying data is in Bold, which can be quite useful to check. You can choose whether or not you want to just search against only species level identifications, or you want to search against the whole database. It's always up to date, obviously, because that's where the data is coming from. It's web-based, again, but you do need an account if you want to get the emails when it's done its analysis, because the analysis can take quite a bit of time. You can't really apply different thresholds like I'll show you in Bold Digger. And it's limited to 50 samples per search, but you can just batch your things into 50 and do multiple searches. It's a bit faffy. I would assume, is Rutger around today? Yeah, there you are. When the API and the underlying code base is refreshed, this limit may be removed. I would request that, but Sujeevan's just like, who's this Ben guy? So if you could request that, that would be quite nice. One of the sort of bigger cons is that the sort of native output is an HTML file, it's a web page, and you can't easily download the top 20 matches of each record in a batch. Like you can do it if you just did one, that's fine, but if you've got hundreds of samples, you can't do it as a batch, but you can get it as a PDF. What Bold Digger does is it literally does the same search you would do in Bold, but it then takes the HTML and converts it into a spreadsheet. Very clever. Procedure, log in, select the database, put in your FASTA format file, select email if you want a notification. So here we go. I'm sure we've all been here before. When you log in, you get to see how many thousands of projects you're associated with. So with BGE, we've got a container project, this one over here. And then within that, each of the partners has their own project. So there's a proliferation of exciting projects there. I don't know what else I was going to say about that. You just log in. You go to the identification thingy, which is that one, I believe. And you pick which of the databases you want to search against. So in this case, I typically just use the all barcodes on Bold. And then you paste it in there, tick, email me the results, please, submit. And then it'll email you, which is just a link to a web page. This is the result from that search. You can see actually here, I mentioned that WASP. There's a bit of a thing. I think my sample names are a bit too long here, so they're just overlapping. But it's come back with, again, 99.75% to that same genus, but it hasn't given a species designation to that. It gives you a graph of like the top match and the bottom match within the top 20, 50, or 100 hits. And then you can scroll down, I believe, and go to one of those records. So here's an example. It's telling us all of the hits here, whether or not they're early release, if they're private, if they're published. Look at that, private, published, private. Yeah, not a lot of published data on Bold, which is not great. Published as in publicly available. And you can also see that there are really good hits, 99.6% here, to species level identified things. So like you could be quite confident that it's probably that species maculata, because then the next species, well, there's a couple of 95s to maculata and then a couple of 91s to other species here. But I would be relatively confident that maculata is the correct thing, and especially if that's what the taxonomist said, you'd be happy giving it that name. However, the top match, 99.8%, doesn't have a species designation, and so Bold has gone along and said, I think it's in the genus, because it's looking at this underlying database, this data set, and it's probably saying, I'm not 100% confident I'm going to give you a species name. I'm just going to give you that genus name. So that's just something to keep in mind when looking at these results. One of the cool things about Bold is that you can also generate a tree quite quickly. So if you were doing one record, you can also download the spreadsheet of those top 20 or top 50 or whatever hits. But again, that's if you're just doing one. But if you want to look at the tree, go up there, click View Tree or download the tree. And what it does is it takes the top 100 hits and it creates a neighborhood joining tree. Neighborhood joining trees are the devil. We don't like them for phylogenetics and things like that. But in terms of clustering to see how close things are, they're useful. And you can see here in that example, that specimen is your unknown specimen. In these trees, these are all black because they are Bold data, the data that's loaded up to Bold. However, if it has sucked in data from GenBank, those would be blue. And the only reason there is because there's different metadata standards for NCBI versus Bold, so that you can see that visibly. But it's nice to see, oh, look, it comes really closely to this maculata clade, and the nearest relative is also maculata over here. And then there's quite a big distance to, hey, look, another maculata. So maculata maybe a couple of hidden species. Who knows? We should go look at that. And then, yeah, I'm not going to go through the whole tree. Yes. Who put the data up? And yeah. So yes, there is a way. If the data is published, publicly available, then you can go and see that record and who identified it and who sequenced it and where the voucher is and all those sorts of things. If it's private, no. And one good thing about Bold Digger, which is the one I'll go on to next, is it can download that public data for you into the spreadsheet. And then it has additional checks that it does, and it'll flag this record is only based on a lot of samples that have used reverse taxonomy. They haven't used morphology, and so it flags those sorts of things. Yeah. But one of the things I'll talk about in version 3 of me is about reference library curation and work we're doing to try and improve the underlying data. Yeah. Yeah. So Bold, well, it gives you kind of essentially, it gives you a confidence, but not a likelihood, basically, where it just says we're confident it's that species, we're confident it's this genus, but we're not confident enough to give you a species. Yeah. So it doesn't give you a likelihood. Yeah. Yeah. Cool. Any questions about Bold? Sorry, I'm probably going a bit fast. So now Bold Digger, my favorite program. Would be nice if it was a bit faster. It also pulls down the top 20 matches, which helps show the underlying issues with Bold. It uses public and private data, but you don't actually get the underlying metadata for the private data, but at least it can match to it. Everything gets downloaded in a really clever Excel spreadsheet. It's always using Bold, so it's using the most up-to-date data. And it flags matches, as I just mentioned, that are on reverse taxonomy if it's just matching to private data, all those. It's got a whole set of flags that come with it. It is slow. I've got it running at the moment. It won't be finished yet, so maybe at lunchtime it'll be finished on our current data set that we've got here. But you can run it unattended, and if it crashes halfway through, you can restart it where it left off. So yeah, and it has a command line version as well. So you can install it. You have to use the command line to launch it, but it's relatively straightforward. You just get in there as an admin and type Bold Digger. You set the input and output folders. You log in with your credentials. This is important. This is the only way I get it to work these days, is you set the batch size to 1, which means it will search one sequence at a time. So it'll search Bold for one sequence, download the HTML result, repeat, repeat, repeat as it goes through your spreadsheet. You can imagine if you're doing a meta barcoding study and you've got thousands of OTUs.
That's another story. We doing that and then it got cut off by
Bold. Yeah, because too many API queries, right? Yeah.
They didn't like it.
Yeah.
The whole university.
Yeah, don't ban your university from Bold, no. When I started using it a few years ago, you could set the batch size to like 50. So I think it's a Bold, I think it's timing out basically on the response. So anyway, nowadays we set it to 1, but of course Rutger's going to get that improved for us. And then the cool thing is it can download the additional data from Bold, so that the stuff that you don't even see on the Bold web page, it now has every single record that it matched in the top 20, and then it goes back to Bold and downloads the original data of who identified it, how they identified it, a link to the record, and all those sorts of things for you.
And guess the top hit as well.
Yeah. And then you can choose how you want it to match. Do you want just the top hit? Do you want it to use the JAMP method, which stands for just another meta barcoding pipeline developed by Vasco Albrecht, and that uses threshold. So it says, okay, and it's quite complicated, so I won't try and explain it because I don't really understand it, but basically it's like if it's over 98% match, it will happily give you a species name if there is only one species name in those matches. If it's above 95%, it'll happily give you a genus name if there's only one genus in those matches. And it goes like iteratively down the route, yeah, which is quite cool. Yeah. That's where you get it from, Bold Digger. Cool logo. That's what it looks like when you're running it. And I mentioned the things like your output folder. You have to have a login for Bold. and your batch size here is where you change it from 50 down to 1. It can do other markers. I've never used anything except for CO1. I think there are issues with some of the other markers, and at least like things like binning and from the bold side, like bin algorithms and things like that. So for what we're doing primarily, CO1 barcoding works beautifully. And yeah, you can use the first hit, you can use the JAMP pipeline, or you can use the bold digger method for the thresholding matches. And the bold digger method is essentially the JAMP one at those 98, 95, 90, and something else. However, it just uses the additional private public data. Yeah. So this is how you would set up a folder. This is how you would check your FASTA file. So a good thing is that it's got a part of the pipeline is it'll actually take your FASTA file and check whether or not there's issues with the FASTA file. Sometimes we put strange names or characters into our FASTA files, and that really screws up the searching. So it does a check before it runs that. This is what it looks like when it's running. It says, this will take a while. It will. Excellent. And this is your output. So in this case, what I've done is I've run through the software, and then I've said, can you also do that additional data for the bold digger criteria matching? And so this is what you would get. This half of the spreadsheet is what you'd get if you just ran it standard mode, but then doing the can you download all the extra data, you get this half of the spreadsheet here, and you can see the person who identified it, the country it was from, sex stage, how it was identified. So was it based on bin matching? Oh, this person used a microscope, that's great. And all that sort of thing. And then a link to the actual specimen page where you can go look at the pictures. Yeah, Glenn.
Is each row there is one of your query sequences?
So yeah, sorry, that's a good question. So these are the top 20 matches for that query. So that's F11, well F11. And here's G12, remember G12 from earlier? And that was the genus, I'm not going to say that, but it was an ichneummonid wasp in that genus, and there's one species level match at 99.74, and it was identified by that person in Germany, interestingly, when the rest of them are Canadian, which is a bit bizarre. But I think Angela works at Bold, so maybe there's an issue with the country for that. But yeah, the interesting thing is you can actually see, oh yeah, there is one species. And I think, if I remember rightly, if I do the JAMP hit for G12, yes. So the JAMP hit has said for this record that we only got a genus level identification later. It says there's a 99.74% identification at that species. The other ones aren't identified as species, so I'm not going to use that as a reason to not give you a species ID.
Can you get more than 20 records for each?
Not with Bold Digger. I think it's just the top 20. Yeah. The command line might have additional options. Yeah. So yeah, so that's the output. 20 for each record with all the additional data here. So if you say, I want the JAMP pipeline to go through those different thresholds, it does that for you. So now this is the first record and what it's matched. Again, it's happy to call it maculata. Remember earlier on Bold, it wasn't happy to call it maculata. It just gave us the genus name. But it's that record it's matched to is a private record, whereas these ones are published records. Yeah. So that's really nice because you can easily compare that to the morphological identifications you have, and then do what I do in Excel is kind of, if I have the morphological identifications in a spreadsheet, I can then do like a match. And so I say, does this cell for species equal the cell for species in the morphological identification? And then I go up the tree, and so then I can filter and say, it's matched at family, but not genus or species. Okay, it's matched at genus, but not species. It's matched at everything. That's great, yeah. Or it's human. And then we do other things. And then there's a third tab, so you probably can't see it here, but that was our run data. That was the JAMP hit that I just showed you, and then this is just the Bold Digger hit version, and it's identical to the JAMP one. It's just added these flags here, and these flags correspond to different things you may want to check related to that match. And I think they are listed here. So you probably can't read that, but flag 1 is reverse bin taxonomy is being used. Flag 2 is there two or more entries with differing taxonomic information above the threshold. So for example, it's matched at 98% species level, but there are 2 species names in those matches. So you definitely need to go check that. And then, yeah, private only, or the top hit is unique amongst the 20 hits. So something's a bit funky going on there. And then something to do with the API correction tool, which I have no idea about. So we'll skip over that. But that is Ben version 1. And if there are any questions, now's a good time. Rob.
How many people are suspicious of you and bothered to do the change?
Well, everyone, Rob.
Everyone.
Yeah.
Even
That's why we don't have lots of dirty data in bold, because everyone's so good at checking before they upload it. Good question. We need more suspicion.
Yeah.
Do you want to start a war?
That's why I'm delegating this to you.
You might have found something that might unite taxonomists for once, which is the hatred of a rating system of their identifications.
With my name, I don't think it's right now, but it's bad.
Yep. So that is a really good idea. And what we're going to be doing in the reference library curation side of the BGE project, at least, is automating some of that. So not necessarily checking people's names, but checking have they used morphology, and then grading records based on those sorts of thresholds. So not people-based. But yeah, maybe there would be a useful thing to say, identify, oh, that person has put 50 records in bold and they are all wrong. So we're just going to take those identifications out of the bin system. They'll still be in bold, you can still play with the data as much as you want, but when it comes to species matching, we're not going to match against those things. Yeah. Sorry, Glenn, I think, then Sarah.
It seems like that hotline is a bit. Do you use it for metabarcode?
The bold digger?
Yeah.
I have, but when it was batch sizes were bigger.
Yeah.
Now it's just...
Like
So there are pipelines that will put the public data from bold and GenBank data together, but not the private data. That is private. You can only access that within bold.
through bold.
Yeah,
Okay.
yeah. And you don't get to see it. You just get to hit against it. That's all. Yeah, yeah. Which is not good.
No.
So there is a big push with BGE to make all of this sort of data open. So I think any European data funded by anything from Europe, there will be a big push to make that open. I mean, we can understand why people might want an embargo period because they're describing new species, and there are people out there who will just say, oh, this is a new bin, I'm going to describe the species having never seen the thing because there's a nice image. But yeah, we do need to push more to open science.
Is there a cutoff for that? Like, 4 years?
So that's something I think's been mentioned, is can we put in some sort of automated embargo, like with GenBank, right? You put it in and it says when your paper's published, it will automatically go live. Yeah, that is a very good idea. Yeah. Sarah, then Roya. Just take the functionality of this and stop doing the API queries. You know, they can do it locally. But bold's not a reference library, right? It's a workbench. If you want to make a reference library, take the data, play with it, and I guess it's the key is to make sure that the decisions you made are transparent, it's reproducible, and the underlying data is somewhere that someone could rerun it later. Because these matches a year from now will be different. Like the records might have different names. You know, there will be newer records in there. So that's one of the things, is it's that snapshotting, right? Yeah, yeah. So that's what we're in the reference library curation side of things. The goal is to take all of the data in bold, run it through a pipeline where we have criteria that I'll go into more detail, but the criteria that give the records various grades, and then certain grades will be handed over to humans to check. And they're like things where this bin has more than one species name. Someone go look at it. Oh, there's 20 of them are one species name, and there's one from a master's student with a different species name. depreciate that record. Now it's nice and clean, you know. But the key is that's great for cleaning the data in bold, but people are constantly putting in new data of varied quality. So what you need to do is, in addition to that, you need to say, okay, here's a snapshot. These are the Lepidoptera barcodes of Europe that we are happy with. They are here for anyone to do metabarcoding to download. And then the pipelines need to be developed where it says, right, I'm going to do an iterative search. I'm going to map against this reference library and give it a gold star if it gets a hit. If it doesn't get a hit, I'm going to go to this other reference library that has worse quality metrics and gets a silver star, and all the way down to I'm going to blast it, you know. But that needs the whole community, I think, to agree those sorts of things. No, those you could, yeah, or you would have versions of them. So you would, yeah. And I think Bold, at the recent meeting we had in Guelph, they were talking about creating a reference library. So they're also recognizing this issue, a reference library of Canadian arthropods for their own internal stuff. And what they're doing is, instead of spending a lot of time cleaning the bins, they are taking trusted records for each species. And so they're kind of going the other approach. And so instead of saying, here's all the data, how much of it can we keep? They're going the which is the best record in this bin? Let's keep that one, and let's just forget about everything else for the reference library side of things. So you do lose a bit of like haplotypic diversity and the gradient across, especially a country like Canada, right? It's probably quite big. But it's probably a quicker way, yeah. Because then you can filter. You can say, right, it was identified by someone in this group. It has lat long, all of these other things, all the photos, trace files, all those sorts of things. Okay, what records match that? Pick one from each species, bang, there's your reference library. Yeah, being private. So new species that are being described, that they don't want to make the data public and like show their hand to the other people is one minor reason probably. A lot of people they are working in their project in Bold, and you can have the option to create your project as private or public. And so you can create a private project, have your master's students do all that sort of jazz, blah, 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 and then just walk away from it and forget to make all those things public, you know. That's the great thing about things like GenBank and journals, is that they require a GenBank accession number for any sequence data, right? So it forces people to make it public. We should also do that for Bold. Like if you are talking about Bold, it should have a published record on Bold, you know, because that's the currency we deal with is papers, right? So. Yeah. Otherwise, no one would stick their data in here. We'd all just keep it on our hard drives. Yeah. Yeah. So there are examples of things like CITES-listed species where you might not want to publish occurrence records and sequence data and things like that. There are other, like Nagoya-related reasons why other countries might not want you publishing the sequences publicly. So yeah, so there does need to be that functionality, but I think the vast majority of that private data is probably people have just forgotten to make it public, being generous. But it's only matching against the public data that you were able to capture. Yeah. So why is your data not published? Give us your reason.
Well, you'd have to ask my colleagues who are waiting to publish data.
Yeah.
Instead of putting it out there,
Yeah.
but obviously there's people sit on their stats.
Yeah. So one thing, I'm not going to name names because I'm being recorded, but a very large national barcoding project that has contributed a huge number of sequences. I was discussing it with the coordinator at one of our meetings, and it came to light that they happily publish all the data on Bold. However, they also do curation of their own data set and update identifications and update a whole bunch of stuff in their records. And those updates don't go to Bold. Great, hey? Yeah. So I was like, well, could you maybe just tell Bold about that and just re-upload all your data to them? Because that's horrible. Now we have two different data sets based on the same specimens. So it's not us, thankfully. We're not that scale. I couldn't confirm or deny that. Yeah, I'm going for Prime Minister next, so I'm just practicing. Yes. Flag it. Yep. So there is a flagging system in Bold where you can tag things, and it's something we'll be using for the reference library curation work we're going to do, where we're going to just flag probably thousands of records as removed from bin or needs manual curation or something along those lines. Yeah, and that is something you can also do. If you have a login to Bold, you can just go and flag records. Yeah.